Great, thank you. So I'm a postdoc in Mike Sheets' lab at Columbia University, and I'm really happy to be here, and thank you for the opportunity to present my work. So my research addresses how the mechanical forces that extend axons, how these are related to the biochemical signals that we've come to associate with axon guidance cues. Now, based in part on some of the work I'm going to present today, I think there's a very intimate relationship between the two. The forces and the biochemical cascades are very intertwined. So I'm going to start off with some older literature that I think really lays the foundation for what I do, and that is that growth cones pull. So one of the earliest reports I could find of this is from Junosuke Nakai about over 50 years ago. And what he did was pretty amazing. He just really looked at his neuronal cultures. And what he saw were what appeared to be individual neurons here, cell bodies, and their axons would contact much larger bundles. And just by looking over time, he saw that they would sort of get deflected towards the cell bodies. And he went on to propose that maybe there's a pulling force on it. Fast forward about 30 years later, and Steve Heidman's group in Michigan State did something a little bit more dramatic and I think very convincing. And what they did was to take a pipette, attach it to the cell body of a neuron, and detach the axon, so it's just the growth cone that's contacting it. And what they saw is that the growth cone would actually pull that pipette, meaning that there's a fairly large pulling force being generated by the growth cone. Now, what I became interested in is whether the attractive guidance cues that growth cones respond to, whether those could be directly pulled by the growth cone. And in particular, I was interested in the family NETRINs as attractive guidance cues. So we all know NETRINs as secreted 80 kilodalton proteins. And work from Mark's lab, who initially purified them in vertebrates, showed that they were both diffusible and substrate bound. So my favorite experiment showing that NETRIN can diffuse is the co-culture assay, where you take dorsal spinal cord explant, and you culture it alongside cells, and it's all embedded in a collagen gel. And so if these cells are expressing NETRIN, you get very robust outgrowth. Now, in order for this outgrowth to occur, of course, is the NETRIN has to diffuse through the collagen, so clearly NETRIN is diffusible. However, what they also noticed during the purification, and I'm just sort of outlining the steps here, is that NETRIN wasn't part of the soluble fraction. It was part of the membrane-bound fraction. To get it off the membranes, they had to use 1.2 molar salt. So that's about 10 times higher than physiological, and it means that NETRIN is binding to the extracellular environment. And so I think a good way of looking at NETRIN is that it can diffuse, but then it becomes very tightly associated. And so what I was interested in is whether that substrate-bound NETRIN could provide those grab holds for the growth cone. And so that's what brought me to Mike Sheets' lab at Columbia. And so his lab is very good at combining a technique known as optical trapping with live cells. And so optical trapping is just, in very simple terms, is just passing a far-red laser through the microscope objective. And at the focal point, you can trap a small micron-sized bead. You can move it around in 3 dimensions, but you can also report the forces that are acting on the bead just based on how much that bead is being pulled out of the center of the trap. So my strategy was to coat this bead with NETRIN and present it to a spinal commissural neuron who are attracted to NETRIN. And so in this video, this will be the center of the optical trap. This is the NETRIN-coated bead, and this is, of course, the growth cone. And what you see is very rapidly, the growth cone interacts with this bead, and you can start seeing it being pulled a little bit towards it. And after about 5 minutes, these forces are going to build up, and it's going to extract the bead from the optical trap. Right about there. And so this video is going to be looping, so you can watch it again. So at the time, we could hold this bead with about 60 pico-Newtons of force. And so what it said is that the growth cone can generate over 60 pico-Newtons, but we had no real idea of knowing what that could represent. And so we could see a little bit of deflection of the axon, but we really wanted to figure out whether this represented a real traction force. So one of the ideas is that maybe it's just some receptor-ligand interaction. So we needed to rule out that possibility. So what we did is we substituted the NETRIN on this bead with BMP7, which of course Samantha showed in Jane Dodd's group as to be a repellent. And what we saw was a very different response. So here you can see the growth cone contacting the bead, but rather than pulling on it, it just sort of collapses. Very different response. So then at least we could say it's more than just some receptor-ligand interaction. So to really determine whether this is a bona fide traction force, what we did is we used an assay that we call a micropillar array. So you can think of this as being a bed of nails, where the nails are made out of polyacrylamide, like what we use to run protein gels. And so just based on the deflection of these tips, you can see how the tips of these pillars are getting pulled towards the center of the growth cone, we can relate that to a very specific force. And when I quantified that from over 18 growth cones, over 2,700 pulls, I found that the forces range between 4 and 15. Now, if we compare this to what we saw on NETRIN, so we were seeing 4 times more force on NETRIN. So that allowed us to say that we were probably looking at an attraction force on NETRIN. So this was really exciting when I got this, because then it opened up the whole possibility that these forces on NETRIN could be regulating the intracellular biochemical cascades that we've come to associate with NETRIN. So that's what I'll be spending the rest of the talk on. This is a recent paper that was just published last month in Journal of Neuroscience. And what we showed was that the intracellular protein focal adhesion kinase is activated by these attraction forces on NETRIN. So work from other systems, primarily fibroblasts, suggests that focal adhesion kinase can act as a Mechano-Transduction protein that would convert force into a biochemical signal. And structural work has the following model based on crystallography, that the firm domain will fold up onto the kinase domain, and so what's perhaps happening under force is that this firm domain will get extracted, allowing the activity of the kinase domain. Now, what I need to point out for the next slide is that this tyrosine 397 site, it's an autophosphorylation site, so you can use it as a measure of focal adhesion kinase activity. And so one of the other reasons why I got interested in focal adhesion kinase is because there's really good evidence that it's important for attracting axons to NETRIN. And so in particular, there are 3 back-to-back papers in 2004. This particular slide is from Wen Chen's group. And what struck me about this Western blot is just how rapid the activation of NETRIN was. So within 5 minutes, they were able to detect NETRIN-induced activation of focal adhesion kinase. So very, very rapid. So it made me question whether NETRIN had enough time. I mean, if it's going to be a mechanical activation, NETRIN has to, you're adding it to the culture media, you'd have to diffuse in, bind the substrate, the growth cone would generate forces on it, which would then somehow lead to the activation of Fak. So what I needed to do is to determine whether NETRIN had enough time to bind to the substrate. So here's where I developed an ELISA-based assay that exactly replicated these culturing conditions. So tissue culture treated plastic, polylysine coated, cultured overnight in culture media, applied NETRIN for different amounts of time, washed it away, and then process it. And what I found is, in fact, NETRIN, we could detect significant binding of NETRIN to this substrate within 5 minutes, and that there's a linear increase thereafter. Now, this was promising, but what I really need to do to test the importance of this binding is a way to prevent it from binding. And here again is where I borrowed from Mark's work again, that when they went to characterize the NETRIN receptors, what they needed to do was to co-apply a small negatively charged sugar called heparin. And that was to show a specific interaction of NETRIN with the DCC. If they didn't have that, it would just sort of bind everywhere. So I tested what would happen when I co-applied heparin, and what I found with these dark circles is that the binding was largely inhibited. So now I could return to the Western blotting assay and ask, what's the consequence of keeping NETRIN soluble? And if we just focus in on this lane here, this autophosphorylation site of Fak, see again, I reproduced that just NETRIN alone will increase the amount of Fak phosphorylation or activation. But when heparin's around, adding NETRIN doesn't significantly increase, has a much lower ability to trigger Fak phosphorylation. So you might be thinking, like I was at the time, heparin might be having other roles other than just keeping NETRIN soluble in this situation. So here's where I came up with a completely different way of going about it. And that was to not block the forces by keeping NETRIN soluble, but block them by inhibiting the ability of the growth cone to generate forces. So I did this by inhibiting myosin 2. So myosin 2 is a motor that pulls the actin retrogradely away from the leading edge, and it's the coupling of this actin to the extracellular environment that's thought to be providing attraction forces, those pulling forces. So I confirmed, using the optical trap with a NETRIN bead, that inhibiting myosin 2, in fact, blocks those traction forces. It completely does it. We never once saw pulling forces above 60 pico-Newtons on NETRIN. So you see here is that the phylopodia that are moving around, They can move laterally, but they don't move backwards. So very consistent. So now, since I had a way of blocking these forces on NETRIN, I can then, once again, return to the Western blotting assay. And if we now just look at these last two lanes here in this Fak autophosphorylation, when myosin 2 is inhibited, I apply NETRIN. Again, we don't see this activation of Fak. And what was really striking about this is with P130Cas, another intracellular protein that NETRIN activates, we did see this activation. And I'll have more to say about that in the last slide. But what I want to get to right now is sort of the third key piece of evidence from the paper, that's the forces on NETRIN that are activating Fak. And so this is an optical trapping assay, where what I do is I bring up a bead to one growth cone, and as soon as it makes contact, I release it. In the other situation, I hold it. In both cases, after it makes contact, I leave it for 5 minutes, and then fix it, and then process it for Fak autophosphorylation. In other words, this neuron is feeling NETRIN that's restrained, whereas this one, there's no restraining force. And then when I looked at the amount of Fak activation here, you see there's a very strong Fak activation when there was a restraining force provided compared to when there wasn't. I should just mention that this is the same neuron here. It's just been rotated 90 degrees. So I think these three pieces of evidence, keeping NETRIN soluble with heparin, inhibiting myosin 2, and essentially comparing this restrained versus unrestrained NETRIN, provide pretty good evidence that it's the mechanical forces on NETRIN that are activating Fak. So one thing I don't have enough time to really go through carefully here, but I at least want to mention, is that we did the converse experiment, and that was to inhibit Fak and look at how the buildup of forces, how the buildup of adhesions was affected onto NETRIN. And what we saw is that indeed disrupting Fak either with a pharmacological inhibitor, PF228, or genetically, disrupted that further buildup of forces on NETRIN. And so this has led us to this model that we're currently trying to reinforce with more experiments, of course, is that we know that focal adhesion kinase can directly bind to DCC. So this is work by others through its FAT domain. And that it could also be bridged to actin through the firm domain. So what could happen upon DCC binding to restrained extracellular NETRIN is that the forces acting on this Fak would extract that firm domain, allowing its activation. Its activation could then essentially reinforce the linkage onto NETRIN, which would underlie the turning process. Now, something I wanted to point out here that we didn't really stress in the paper was that I view this more as a checkpoint and not as an initial event to NETRIN. And that's largely based on when we inhibit myosin 2, we still see activation of other signaling components. A few slides ago, I mentioned how P130Cas, another intracellular protein, is activated. And so the way I view Fak is the growth cone coming along, it sees NETRIN, has some initial cascades, and then the growth cone asks, is this something that I can build an adhesion force in order to essentially turn the axon? And that's where I believe Fak is involved. Another important thing to mention with focal adhesion kinase is that work by others, particularly Tim Gomez's group, has shown there's a general trend of Fak activation to be activated in a chemoattractant setting, and for its inactivation to be associated with our chemorepellent setting. So I think that combined with evidence that just about every guidance cue can bind to the extracellular environment, I think this could be a general mechanism. And one more thing, just because I have a minute left here, I think this could even go further than axon guidance. I think in this meeting we've seen that axon guidance cues are involved in synaptogenesis. Paul Brigman had a poster the other day showing that myosin 2 is important for synapse function. There's also work by others that have shown that. And I think that this Mechano-Transduction could be a good way to get a very specific, quick signal across the synapse, or that. So I'll let you tear me apart now. Oh, wait, I should just thank everybody. So Mike Sheets, okay, this is my supervisor, Mike Sheets. He's just been phenomenal. He's long history in science, and he's just this year he started getting a lot of recognition. He won the Wiley in Alaska for his work with kinesin. Nicolas Biet is a postdoc in our lab. He's the one who developed those polyacrylamide pillars. He's starting up a lab in Brooklyn College, and this is a reference if you want to know about those polyacrylamide pillars. Shan Zhang was instrumental in the P130Cas and Fak studies. She's starting up a postdoc in Alan Hall's lab. I've received funding in the past from the Canadian Institute of Health Research, and more recently from the NIH in the form of a K99 and other funding from a PN2 and an R01. Thank you.
Thank you, Simon, for a great talk. Questions?
Great. I can repeat it.
So I'm curious. So soluble versus tethered netrin.
Yes.
Do you think there are two different physiological responses, or are you suggesting that when we're putting soluble netrin into our in vitro assays, we're getting an artifactual? I mean, that's a non-physiological response.
Okay, so so one of the things that I showed there with the ELISA is that netrin very rapidly becomes associated with the substrate. And I've also seen it in the pipette assay. If I reproduce essentially that spritzing of netrin, I can see it become associated with the substrate very rapidly as well. And so I mean, it's very difficult, and I've had conversations with a lot of people on how to rule out the possibility that soluble netrin can guide axons. And it's very tricky because if anybody who's worked with netrin and purifying, it's very, very sticky. And you can do tricks like taking off its C domain, which I do in the paper, but it still very readily will bind to the extracellular environment, not as well, but it still will. So my view is that it's only substrate-bound netrin that's functional, but I think there's room. Exactly. Axons don't grow in a substrate in like free media. They're not trees, they're vines.
Can you say a little bit more about the coupling and force generation mechanism? So the coupling to the actin itself and what you're thinking there, and is the force, there's some indication in other cell types that force at the plasma membrane would be generating a stiffening of the underlying actin cytoskeleton, and is that consistent with the motile forward movement of the growth cone or its ability to grab and
Okay, so what I think you're
generate
saying
force?
So particularly work that's actually done in our lab is you can, when you apply forces on a bead, like a fibronectin-coated bead on a fibroblast, you see a stiffening. You see like, and I think this is what you're referring to, right? And so the idea behind that is that you're sort of forming an adhesion, right? And that's why they're doing those experiments, so I'm not quite clear. So the underlying actin, yeah, definitely. I feel like there's an adhesion being built up, and adhesions are what grow out actin, right? I'm not sure if we can talk more about that.
Hi.
Hi, Allison.
Have you ever tried applying a soluble repellent to see if the soluble repellent can reduce the traction forces that the growth cone is exerting on the netrin bead?
So the closest thing, I even showed it here, was the BMP bead, where I bring it up with the optical trap, and then...
Right, but have you tried putting a soluble repellent when a growth cone is contacting a netrin bead to see if the soluble Q is enough to signal and block, for example, the netrin-dependent Fak phosphorylation? I'm just thinking about in vitro, could a soluble repellent outcompete the netrin attraction in that kind of way?
You see, again, so I view just about every guidance Q as being substrate bound. So it would be just coming up with a repellent that would be soluble in that situation. But and the short answer is no, I have not done that, and that seems like a great experiment. Paul.
I'm wondering if you can really assume that the generation of traction forces and the pulling is responsible for the turning, because when you couple to the cytoskeleton, you affect the retrograde flow. And as we talked about previously, what that can do is allow the invasion of microtubules. And so it's not easy to separate out those two things. So have you done anything to try to...
So I have actually had some time to think about it since I've been at your poster. And I think, so your model is microtubules are essentially what's important for turning the axon. And so if you get rid of actin dynamics, the microtubules will push forward and can essentially form the adhesions that would further extend the axon. Is that roughly how you view it?
Stabilize the adhesions.
Stabilize the adhesions. Okay, so the the reason why I don't like that model is that we know, as you've seen with myosin 2 inhibition, that they're less selective, right? So with myosin 2 inhibition, you get invasion of the microtubules into the leading edge, and growth cones aren't able to choose the right path. So the way I view myosin's role and actin's role more generally is to evaluate the environment that's been presented to the growth cone and saying, what's the best one? And when you figure that out, you can sort of move yourself forward and let the microtubules through. But yes, I don't think there's a clear-cut experiment.
I guess I would argue that you get selectivity by the ability to form a adhesion site selectively slow the retrograde flow in one area to allow microtubules have a greater chance to extend in that one area. And that is how you get the turning response.
Okay. I guess we're going to need to talk about that a lot more. Yeah.
So we have 2 more questions, but quick. So
Yeah, very quick, yeah. So in your movies, the beads are actually being pulled not at the tip of the filopodia, it's more kind of close to the base of the filopodia, even in the lamellopodia area. So as we know, the tips are very sensitive. So it is possible, and the possibility is that when axons, growth cones growing, you add more membranes, and then you also have membrane flowing back.
Oh.
So it is possible the beads is actually being carried by the backward movement of membrane flow. Because that's
So
possible.
I don't think it would be, membrane flow, the membrane is very fluid. And so Mike, on top of a lot of other things, my supervisor has had a lot of experience with membrane dynamics, and it's a fluid thing. If you have a bead, you can get it tethers, and you can just sort of move it around. There's no forces acting on it. The forces come from the actin cytoskeleton or the microtubule network as well. But the membranes themselves.
Actin is not touching the beads.
What's that?
The actins are not touching the beads.
The actin is being bridged to the bead, is the way I view it. And so if you, the phospholipids in the membrane are movable, right?
okay, one last question. Quick.
In the polyacrylamide pillar assay, it looks like the axon is pulling on the pillars as well.
Yeah.
Have you ever put a bead next to the axon to determine if the axon is able to pull the bead?
Okay, so that's a great question. So one of the things, I have this other video, and it's actually the one that's part of the science paper. And if you look very carefully at it, there's a lot of things happening with the forces when you start looking at on the pillars. When vesicles go by, there's this big you can see contractions around the environment. I would imagine there would be. If you let these videos go, like after the bead is pulled out of the trap, the bead goes all the way back down to the cell body. So in other words, it has to be being passed off from the actin network probably to the microtubule network. There's not a whole lot of actin in the axon, and it's being transported towards the cell body. So there's definitely forces on it. And yeah, so my short answer is yes, I think there's forces being generated on there.
Okay, thank you, Simon.
Great.